Hi, everybody. It's so nice to see so many people join us. My name is Carmen Trupek. I direct scientific programming for Global Genes. Hopefully, many of you know just a tiny bit about Global Genes. We are a nonprofit umbrella patient advocacy organization, been around for almost 17 years, supporting broadly the rare disease community. Um, Global Genes has a research program, a research platform called RareX. And RareX, as the research platform, is a place where patients and caregivers can come and collect research grade data that is owned by them. So we were really, the platform was founded to provide a way for rare disease organizations to collect really robust research grade data that's widely available. It's not owned by an academic institution or any particular one study, but is widely available for research to really like push forward the pace of research. So we've been collaborating with the Pompe community, with Ryan and Heather and Paloma and Anne, and many other advocates in this space. Amanda Holthouse also did some promotion for this, and so we've been collaborating with a number of individuals in the Pompe community to build that out. So as Heather and I were talking a couple of months ago, we were talking about like what are some of the big questions and needs in the patient community? Because if this is going to be a patient-owned, patient-led initiative, let's make sure that everything we do is really listening to the needs of the community and helping to build and support on that. And what Heather shared with me is that there are so many questions about gene therapy. What is gene therapy? What other types of therapies are related to gene therapy but different? Are all gene therapies kind of a one and done? What are the risks associated with gene therapy? We do not promise to answer all of those questions today. Today, our hope is that we can really make this an open meeting, an open conversation. I have provided, or I have created a few slides that I'm happy to walk through to provide some overview, including sort of a snapshot of what's being developed in Pompe right now and what's on the horizon. But in full disclosure, right, I work for a nonprofit. I don't work for any of those companies. And if I get something a little bit wrong, I encourage the representatives of those companies to correct me. But it's not meant to be in depth of any of those, but really big picture so that we can foster lots of conversation and figure out what are the questions that we don't have answers to today, but that we commit to getting answers for you. By background and training, I'm a clinical genetic counselor, and I've spent many, many years working with different rare disease communities. And part of that work has included really identifying, engaging, and supporting individuals with rare diseases as they navigate clinical trials and clinical trial eligibility. So I do have some expertise to draw on to be the one to present some of this information to you. But I'm not a doctor and I don't work for any of those companies. So we'll get you those answers when needed. That was kind of longer than I intended. Caitlin.
Hi, everyone. Happy Friday. My name is Caitlin Hobbs, and I'm the Senior Manager of Community Engagement at Global Genes. I work predominantly on the Rare X research program, as Carmen mentioned. I really am kind of the main point of contact for all of the patient communities and patients. And I just wanted to thank you all so much for your participation today and really excited for this conversation we're about to have. So I'm going to pass it back over to Carmen.
So first off, I would love to not just keep talking, but to ask if like right off the top, and if you want to get on screen or not get on screen, we don't care. If you want to put it on the chat versus like put up your hand, like actually or virtually, we don't care, but would love to know what inspired you to join us today? What questions do you have? What are your worries? What are your hopes or excitements? Around gene therapy and other gene-based therapies.
I'll say something.
Great.
I just kind of want to know where we are with this because we've been hearing with it for so many years, and this study started and then it wasn't. And I mean, I was evaluated for one study and rejected, but I just kind of know where we stand and what the future is. You know, it's everything's in development, but what's going to actually happen and when? Time frame.
Great. Great question. I'm going to cover a little bit of like what's happening right now in terms of what's in clinical trials. I'm not going to have all of the answers about timeline, like if the clinical trial is closed, when is the next one going to open? When do we expect results? But that is something that I think we can invite some of the representatives from those companies to come and tell us what they can. They can't always answer those questions, but we know in conversations with them that they're willing to come and talk to us. So we'll answer some of those questions today and some of them in follow-up. Other questions?
Carmen, you and I had a conversation a couple days ago or maybe last week about some of these treatments that are maybe in the pipeline, where you were talking about like the difference between gene therapy and gene editing, and there was another one you mentioned. Can you talk about those so that patients have an understanding of what they are and what that means, and maybe helping you to make an informed decision about whether or not that's something you would, maybe a clinical trial you would want to participate in?
Mm-hmm. Maybe I'll just go ahead and start, and I'll share the slides that I put together to tee up this conversation and talk about kind of where things stand right now. And totally interrupt me as we go. This is not a formal presentation. So interrupt me, put questions in the chat. Heather, you can monitor what's in the chat. So let's just do that. Let me share. I have two screens. I always start this way and then do this to make sure it pops up on the right screen. Okay. So again, this is just a little primer that I put together. It will not answer all of your questions. Okay, so some people, some academic institutions have sort of gone to using this term genetic medicines. And that is because there are different types of therapy that don't all neatly fall under what we would traditionally call gene therapy. So I have put what I would call kind of the big four here: gene therapy, RNA therapy, gene silencing, which is actually more than one type, but it's kind of a category, and gene editing. I think everybody here has heard of gene therapy, and probably everybody has heard of gene editing. You might not necessarily have heard of the others. Big picture, gene therapy is often called gene replacement, but it's really not replacement. So for any genetic disorder, there are, this pop-up is getting in my way, move it. For any genetic disorder where there is a genetic variant or a genetic mutation in the gene that is causing that gene not to work appropriately, if gene therapy is a reasonable approach, you deliver a working copy of that gene to a particular tissue for that person. You don't actually swap it out, right? You're not taking away what's there. You're just adding. So gene replacement, I think, is a misnomer. It should really be addition. So gene therapy tends to work very well, broadly. It tends to work well when you have a genetic condition for which the gene isn't working appropriately. The gene is normally supposed to make a particular protein. And if you were to supplement, if you were to add in a working copy of that gene starts making the protein, and that's going to provide significant benefit. RNA therapy, and I'll show you really quickly in a moment, like the difference between DNA and RNA, is also designed to produce more protein. So there are some types of approaches that are really intended to be in addition to create more protein. There are some approaches to therapy that are actually intended to do the opposite, that are intended to block something harmful. So for the most part, in Pompe, what we're talking about as the problem, of course, is that you're not, if you have Pompe, you are not making the appropriate level of enzyme that you need to break down glycogen. So we need to add back in the capability to do that if we're talking about gene therapy. We don't need to silence typically something that's really harmful. So you won't see a lot of like gene silencing approaches for Pompe. Gene editing is fundamentally different than gene therapy or an RNA therapy because you're not adding in like a completely new gene, but instead you're doing essentially like a Microsoft cut and paste. You're essentially doing this genetic cut and paste to modify the existing DNA. Now, in order to do that, of course, you have to deliver the mechanism to do that. And so sometimes that can be through, a virus, the way that like gene therapy uses a virus as the transport vehicle. You could use a transport vehicle like that. But it's quite different. Gene editing came around a lot later than gene therapy. So in general, it's a little behind in terms of science, but is getting a lot of traction and is a viable approach for Pompe. Okay, let's take a quick step back. So when we think about how we modify genes, our bodies are made up of trillions of cells. Every single one of those cells contains 23 pair of chromosomes. Those chromosomes contain all of our DNA. The DNA is continuous, and there are little segments of it that are the specific gene, right? Those segments, the gene, those are made up of these 4 bases: adenine, guanine, thymine, and cytosine. And that probably sounds familiar from like 100 years ago in high school biology. That's what it feels like to me. But AGTC should seem more familiar from your genetic testing, right? So here's some example variants, or what usually gets called mutations in Pompe. And if you look at the last part of it, you'll see all of them have either A, G, T, or C as part of the like mutation name or the variant name. So in this first one where there's supposed to be a T, that got swapped out with a G. In the second one, there's a deletion of a T, right? In that third one, there's supposed to be a C, but instead there's a T. And all of these tiny, tiny mistakes can really lead to that gene no longer being read properly, which needs to happen in order to make the protein. Right? So here's that gene again, here's that section. When that gene gets read, the first thing that happens is it splits apart the 2 strands into a single strand, and it creates the strand of RNA. So the RNA is really like the blueprint. That's what really truly gets read to create this like very, very complex protein. The vast majority of enzymes, including acid alpha glucosidase, which is the enzyme needed to break down glycogen, the vast majority of enzymes are proteins, including this one. So this is sort of the big picture mechanism. So you can imagine then, if you think about this, we could attack the problem up at the gene level through gene therapy. Or we could attack the problem at the RNA level by like putting in a working copy of that RNA strand, the blueprint, right? Like if there's something wrong with the blueprint, let's just pull in a new blueprint. Or you could correct the problem at the protein level. This is what's happening today. The standard of care in enzyme replacement therapy is you say, well, I'm just not making enough of this protein, so we're going to deliver the protein, which in this case is an enzyme, through enzyme replacement therapy, right? There are some future forward-looking ways of thinking about therapy where you could also look at this final step. You could say like, well, if the whole problem is the buildup of excess glycogen, could we prevent that problem without actually putting more enzyme in? And you could do that too through something called substrate reduction therapy, which just reduces the amount of glycogen that has to be broken down. Heather has raised her hand.
So going back to that previous slide, where you had the information about like a few of the mutations that have been identified.
Mhm.
So when I'm looking at this and and then thinking about the number of mutations that have been identified in Pompe, um, and I don't know, maybe this is a silly question, but it seems like is gene therapy or gene editing, would these things be specific to patients? And I know they're
That's a
not.
super good question. Like, that's not a silly question at all. It's a super good question. So, gene therapy is designed to deliver a full working copy of the gene all the way through. So it doesn't matter where the genetic variant occurs, where your mutation is, doesn't matter. Because what's going to be delivered is a whole working copy of the gene, which creates its blueprint, which creates the protein, right? So the nice thing about gene therapy is it's completely irrespective, independent of the specific genetic mutation. There are some approaches that are genetic mutation specific. So when we think about like gene editing, well, if you're going to cut and paste, you have to know exactly where the variant is to know to go in and cut that out and replace it. So a lot of the early work in gene editing has been done in rare diseases where there are recurrent mutations that are like really, really common in a particular rare disease community. There's lots of research and discussion in the research community about how to make gene editing like higher throughput, where you could essentially like design a clinical trial where it's the same mechanism of delivery, the same gene editing approach, but each one has like a little tweak in the code, right, to go after different mutations. Um, so I think that's a little bit forward thinking right now, but hopefully not too far off. Have you seen other questions come in? It's not easy for me to monitor the chat in the view I
I'm
have.
not seeing other questions. Orion had put something in, but I don't see it anymore. Brian, who is from the Columbia group, says that Fernando was at the Duke conference back in April. And yeah, he and his caregiver attended, and he said that Fernando reported that patients in the U.S. have a better quality of life or health, and that they're interested in learning from the U.S. so that the new enzyme in gene therapy can reach Colombia.
Yeah, I think that's a big goal for all of us. It's probably outside of the scope of today's conversation, but important for us to put on the roadmap of things that we talk more about. There's this long road to getting something like a gene therapy approved. And then once you get to approval, then you start thinking about access, right? And of course, you actually think about access before the approval date. But once you know, like, oh, this is probably going to get approved, and let's say that's in the United States, and shortly after, so the FDA approves it, and then shortly after, it's in Europe, and the EMA approves it. You know, still, that can be a very long road for a lot of other countries to get it approved. And what does that mean? So, again, that's like kind of beyond my scope, but something I think that we should think about, Heather.
Um, Dawn wanted to know if this is IV. Will this be IV delivered or how is it? How will it be
I have a
delivered to
slide on that. I'll show it in
patients?
just a second. Delphina?
Okay. Oh,
You're on mute.
you're on mute.
You're
You're
still
on
on.
mute. Let's see. I'm trying to see if I can unmute her, but I cannot.
Hi.
Oh,
Oh,
yeah, there you are.
Hi, how are you? I'm from Argentina.
It's good to see you.
Yeah. There's any difference between gene therapy for infantile Pompe disease or late Pompe disease? My baby has Pompe.
mhm. I think that like the design of the gene therapy is not different. The way that we approach clinical trials, at least in the United States, the way that we approach clinical trials is a little bit different um, because of the inherent protections of children. And so here, you know what? The last two questions, I think let's go to this. So what's in the works for Pompe disease? Like I'm going to partly answer your question through this. If you look at this, and I really apologize if I missed something, but I tried to like really scour what's on clinicaltrials.gov for what's currently listed there. So if there's anybody from pharma who's on here who feels like there's something that is currently in trials that I have missed, like please put it in the comments and we can add it, we can follow up. But in the United States, there are these 3 different clinical trials right now for gene therapy, right? So most of what I have on this chart is gene therapy. They're all right now in phase 1, 2. I think they're all currently closed to enrollment, which is actually good news. It's actually really good news. Like the thing we all should want as a larger community is for these trials to fill relatively quickly, because then we'll get to data more quickly, right? That tells us like, is it working? Does it seem to be working? Is it safe, right? And can we move forward? So traditionally in clinical trials, you have phase 1, phase 2, phase 3. Phase 1 is safety only. It is not designed to determine whether or not it's actually working. Phase 2 is small studies designed to determine, like, does it seem to be working? Is it still safe? And then a phase 3 study traditionally is like a large study in a larger population where you're like, your criteria for who can be in the study is broader. You really want a wide range of experience of a sort of disease spectrum so that you can determine, like, does this work across lots of people with different kind of varieties of this disease, different ages of onset? In rare diseases, we almost always see these 3 phases condensed into 2. You see a phase 1/2, and you see a phase 2/3, right? Right.
I'm sorry, Carmen. The Sparks program is no longer active, is what I was just informed.
Okay. You know what? Yeah, it's not updated that way on clinical trials from what I could see, but I had
Okay.
heard something about that at the meeting. Okay. All right. So, these other 2, right, in the phase 1, 2, right now we are all awaiting results. Right now we are all like crossing our fingers and hoping that the early data, and what typically happens is that there's a long phase of like, oh, we're going to follow patients for 4 years or 5 years in this study. But the FDA doesn't always make you wait until the very end of that before you start really planning and developing your next phase study if they're really good, strong early results. So right now we are all awaiting these early results on these couple studies. And we might have some input from Astellas or Ask Bio on that. And if not, we can do that in a follow-up meeting where we bring them in and have them say what they can say. But you see that these are for adults with late onset Pompe disease. We all want gene therapy to work. We all want to see clinical trials where children are enrolled. We all want to see IOPD included. But it does not surprise me, and I have no personal insights into like these companies and their conversations with the FDA. I have none. But it doesn't surprise me given the last, 24 years working in rare disease and working around a lot of clinical trials that they're starting with adults. They're starting with sort of a kind of a lower risk population in a phase 1, 2 study. That's pretty normal.
Yeah,
go ahead.
I have a few things in the chat. The first is just a comment that Arrow and Shinogi, the former Maze asset, are both working on substrate reduction strategies, and both have initiated clinical trials which go into the other types of
I
The
have
things that you mentioned.
this.
I'm sorry. Uh, sorry. And then so that was just a comment. And then I have a question here. If gene therapy adds a full working copy of the GAA making gene, does the original gene continue making the non-glycogen binding GAA? If so, is there any effect of the non-binding GAA that we should be aware of? Sorry for the long question. Happy to clarify, she said.
No, that's okay. So generally, I'm going to say generally, and then again, we might have to get like more specifics from some of these companies and the way that they think about it. Generally, in a disease where you're supposed to make a particular enzyme and you're not because of a genetic mutation, or in this case, two genetic mutations, one inherited from mom and one inherited from dad, right? So in general, what we see is that either that person makes less enzyme than they need, or they make kind of a not so functioning enzyme, really poorly functioning one. But it doesn't do overt harm, like making a teeny tiny amount and then adding to it a higher amount is fine. Also, keep in mind that in gene therapy, it is very rare, broadly, in gene therapy is very rare that the gene therapy actually ends up generating the quote unquote normal or like average 100% level enzyme. That's actually really uncommon. But the good news is that you get therapeutic benefit in most rare diseases where gene therapy works without having to get to 100%, right? Think about like everybody who's a carrier for a recessive genetic disease is probably only making about 50% of what someone who's not a carrier is making, but they don't have Pompe, right? So like 50% we know is enough in almost all recessive genetic diseases. So if the gene therapy is delivering something that is getting you maybe 40% and before you were making 5%, like you're getting close to a carrier, but you're not getting like wild, crazy high amounts. It's not harmful.
I have another question. What area of the country is research being done? Duke University, Florida, California, are any particular age groups being targeted for the research?
So for every clinical trial that is active, it should be listed on clinicaltrials.gov, which is exactly what it sounds like. I don't recommend that you all go and scour it right now because I worked hard to do that, and we're getting feedback live from some of these, company representatives right now. But at any point when there's an open trial, if you go there, you will see what the sites are. So obviously Duke is a really big site. There are other like big academic centers that are participating in Pompe clinical research, but often there may be a clinical trial that is like at, 3 sites, and a different clinical trial that's like at a different 3 sites, because realistically, they want, to the extent possible, to draw from different areas of the country, from different patient populations, to enroll their studies faster. But there's not a master list of like, here are the 5 centers where all clinical trials are and will ever be. It really varies based on the trial getting started. And typically, once, like, let's say Astellas and AskBio both get good interim results and they both move forward into phase 2/3 clinical trials, which we all hope is going to happen. It's very common that when that happens, they'll have more sites than they did before because they're going to enroll more people in the next study than in the first one.
I have another question. What about children that are in between infantile and late onset? what category do they fall in?
I think maybe that's not something I can probably quite answer. That might be something we want to do in a follow-up, particularly in consultation with a couple of these companies. And maybe like a good topic can be to have a couple of them, like to have Astellas and AskBio perhaps talk about how they develop inclusion and exclusion criteria, who can be in a clinical trial, like how are those criteria developed and what how do they make those determinations? I think that would be a really good thing to have them talk about.
I am including in the chat to everyone. This is a link from Arrow and their clinicaltrials.gov information. So there's a link there. And this is just, it says it's study details for safety. So it's a reference for everyone.
Perfect. Perfect. Yeah. I mean, I wanted to make sure to talk about it. Like if we go back to this, right? And you think about down here, this pretty but obviously complex protein example that I have here. If you think about, this as the enzyme, an enzyme replacement therapy is here. Like, I created that chart that we were just looking at, thinking about these really gene-based therapies, which would fall under the gene and RNA level. But I did not want to forget about, this piece in the sort of pink purple over here, which is, are there other approaches to getting rid of excess glycogen? And if we can inhibit the production of too much glycogen to begin with, which is what substrate reduction therapy really is, then you're just approaching the problem a different way. So yeah, that's great. And I don't know, because I didn't dig into it for those, because I was mostly doing the gene-based therapies today. But maybe somebody also from Shionogi can tell us where they're at with regard to clinical trial status.
And that actually kind of leads into this next question. Marty wanting to know how you can find updates on the progress.
Mm-hmm. I mean, I feel like something that we should just commit to. Like having a central place to be able to kind of stay on top of it, at least like quarterly, right? Because it is cumbersome to try to read through everything on clinicaltrials.gov. So let's take that as an action item and we'll follow up with everybody about how we're going to do that. Okay. Yeah.
Did that mean only why immunomodulation?
I don't think I quite got that.
Jose, we didn't really hear what you said.
Yeah, did that indeed only why immunomodulation?
I still didn't understand. I think. Mm-hmm.
Is there someone that can maybe help us? I think he's speaking Spanish. Is there someone that can maybe help?
Yes, me, Delfina. A ver, let me see. Do you have the question on the chat? Mhm.
Jose, if you put your question in the chat in Spanish, Delfina will translate it for us.
He said, does this therapy require immunomodulation?
Right. So this is a big question for a lot of people with various therapies is, will I, or in a future study, my child have to be put on like an immune suppressant, right? So That's a level of detail that I don't think I'm qualified to answer. And so let's pull that out as one of the questions that we really want to get the company representatives to talk about, and not just talk about like if you have to, but like what are the risks associated with that? Why is it important to do? Is it important to do in every person? Is it, dependent upon something like CRIM status? Like, let's get some of those answers.
Do we have any other questions?
One of the things that
Comments?
Heather and I talked about, oh, was there another question, Heather?
I was just saying there were questions or comments.
I'm going to say this while people formulate comments or like other questions. When Heather and I last talked, we were talking about how it's sometimes people will look at other genetic diseases as examples, and it might be helpful to look at one of those. And so I did put this together that we can look at, and I think it's informative in a couple of ways. So SMA stands for spinal muscular atrophy. SMA is as genetic diseases go, SMA is a really common disease. It's more common than Pompe for sure. It's been on the newborn screening panel across the United States for some time. If you look at this top picture on the left, this little baby has so little tone, so little muscle tone that they can't even hold up their head. This is very normal. So almost all of these children without therapy will die before their second birthday. Therapies for SMA over the last 8 years have been completely transformative in this disease. This little girl over here is essentially living a pretty normal life. And that is just unbelievable. For somebody who's worked in clinical genetics for now almost 25 years, like it still brings tears to my eyes, even though this happened like 8 years ago. Like it's really quite incredible. But I think when we look at what therapies have been approved and how they're being used today, it's really informative as we think about genetic diseases broadly. So the first one that was approved is a drug called Spinraza. Spinraza is actually an ASO therapy. So an ASO, which stands for antisense oligonucleotide. So it actually like corrects this incorrect splicing defect. Now the thing about SMA that's very different than Pompe is that there's like a duplicate gene, like almost like a hidden sister gene. So there are these two genes, SMN1 and SMN2, and SMN1 is like the big brother gene. It does most of the work, but there's this SMN2 gene that kind of boosts extra production, and so it can be like utilized. So that was first developed. Now that was amazing, like had tremendous benefits, but it requires loading doses, which are like 4 doses over a period of 6 weeks or something like that, or a month, something like that. And then it has to be delivered once every 4 months, and it has to be delivered intrathecally, so through essentially like a lumbar puncture. So this is quite invasive. But if it's going to save your child's life, it's like, it's a no-brainer to do. So gene therapy came along, and this gene therapy, many of you may have heard of because it got lots and lots of press as the most expensive drug of all time when it came out, right? This was the $2 million drug, Zolgensma. This is a classic gene therapy. It is delivered by IV once and only once, ever. It is meant to be a one and done therapy. However, you've all read ahead as I'm talking by now, and you've seen on the bottom that although gene therapy is designed to be a one and done, there are some SMA patients who have had significant disease worsening some number of years after their initial gene therapy who are now also being supplementary treated with one of these 2 other medications. And so I think this example is informative because we have to keep in mind, like, there's so much hope in gene therapy, but we also have to temper it a little bit. And it may be that like thank goodness there are multiple companies developing multiple approaches to treating this condition, because over time we might need combination therapies, or we might need backups for certain patients. And then there's this RNA therapy as well, that's an oral medication. So, if we could get to the same thing for Pompe, where maybe we have a one-time IV administered gene therapy, but there would also be this substrate reduction therapy that would either be, intermittent with a hospital visit, or it could even be oral. That would be a pretty great goal.
I have two more questions. Are there any studies geared towards carriers? And for the gene therapy for SMA, what approach was taken? AAV. Was it AAV?
I'm pretty sure it was AAV. Probably someone else in here either already knows or can look it up while I've got this view in front of me and can't actively search. I think it's an AAV though.
And then what about studies for carriers?
Oh, sorry, studies for carriers. And does this mean just broad research studies, or therapeutic studies?
She just said studies. And I know that's a question that comes up sometimes in the Pompe community, that people who are carriers report having symptoms, and trying to find someone who's doing research on that.
Yeah. And I think that that's not uncommon in rare diseases where, of course, most of the focus is on people who are affected, adults or children, who are very severely affected. But we don't want to forget that, if there are carriers who are experiencing some symptoms that could be treated, we at least want to systematically learn about those and learn about the need. I do think that this is a place where, here I mentioned right up at the top. Oh, sorry, I'll talk about the naming in a second if you haven't voted yet. But we do have this program that we are doing, this patient-owned, patient-led research program in collaboration with the Pompe patient community. And actually, this is a place where we designed it in such a way that carriers can come in, especially let's say that you are the parent of a child who's affected. Your child is a patient, but actually you are also kind of a patient. It's really a participant. You're both participants in the platform. But the way that the platform asks questions is very, very much through branching logic, so that you only ever get questionnaires that are, or at least theoretically, sometimes we make little mistakes and we have to fix them. But for the most part, you should only ever get questionnaires that are applicable to your experience. So when you first come in there, if you're a carrier and you get this very high level head to toe questionnaire, and it asks you, do you have any problems at all, let's say with your vision? Do you have any problems at all with your hearing, right? Do you have any problems at all with gastrointestinal function? So it's just very high level, ask you these questions. If you go through and you're like, I am healthy as a horse. No, 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 no. You will not get any follow-up questionnaires about symptoms. But if you say yes, I have some hearing loss, and yes, I have some GI dysfunction. And you know what? I also have this persistent brain fog, or maybe I have some muscle weakness that is really undefined. If you say yes to one of these things, then you're going to get a follow-up related to that. And we really want to use that to make sure that we are, to the best of our ability, really capturing that experience of carriers and sharing it with the research community. I do have here, while you're listening, if you want to just grab this and help us name it, Pompe Disease Data Collection Program is a horrible name. It's not a real name. It's a placeholder name. We need a better name. Whenever I was showing you that slide on the clinical trials, they all have names, right? The Fortis study, they all have these names. So we've gotten input from a number of different advocates in this space on some potential names. And I thought we would have a name by today, but it's neck and neck between a couple. I won't tell you which ones because I don't want to influence anyone. But if you just grab this and vote, we're gonna try to close it so that the next time we all talk, we're gonna have the name and we're gonna start to use that moving forward.
And Caitlin just put the link to vote in the chat. So you can use that, or you can use
Oh yeah,
the
you can click on
QR
it if
code.
that's easier. It's probably
Yeah,
easier.
you can use either one of those. Do we have any other questions or comments? We're really interested in hearing from the community. And this is a series. So we have 2 more sessions planned. And we really want the community to kind of guide how we do this. And so we're very we're happy to receive any input or feedback that you have.
Having just popped out of screen sharing, I'm like, oh, 29 things in the chat. Did we get to all of those?
I think we did. So what do you mean by can we have them outside of business hours?
I'm like half paying attention because I'm trying to work and I would like to be able to pay attention.
Oh, no
So
worries.
several things. One, this got recorded. We'll share
Yes.
that. 2, I am very happy to share my slides after I get feedback to make sure that, like, if I said something wrong or one of these companies is like, please remove my name, like I'm going to make a couple of edits. But after that, I'm happy to share these slides with Heather and she can make them available.
Okay, thank you.
And once the recording is edited, Pompe Alliance will share it on our YouTube channel. And we always put the link out so people can access the information. Did we get to everybody's question? I want to make sure. I went back through and I think we got to everyone. I want to just make sure that we did before we end for the day. Or if you had a comment.
I would love it if other people could just pop in there, like, in this series, who do you want to hear from? Do you want to hear from people who have been in a clinical trial and hear their personal experience? Do you want to hear from the companies that have current clinical trials on, like, what you can expect next? And is there anything they can say about the next study and what's planned? Do you want to hear about clinical trials in other countries and when you can expect to see them there? Like, what are the things you want to hear in the next 2 meetings?
Also, and that's a great point, Carmen. And also keeping in mind that not all, while a lot of the clinical trials are around gene therapy, not all of them are associated with gene therapy. And so your experience in a clinical trial, of course, may be different depending on what it is. But there are other aspects of clinical trials that might be the same, and hearing from people maybe who have participated in different types of clinical trials could be helpful. Delphina said all of those.
All of those things I just threw out. All of them.
I think it
Yeah.
would be great to have experience of a patient out of a clinical trial. I doubt that someone from the industry side will be able to talk about this because often these things are very confidential.
What I've heard from the companies that I reached out to is that because we're asking them and we're inviting them, they would be happy to speak, but they probably can't say all the things we want them to say. They probably can't say, like, I expect interim results on this date, and we're hoping to start the new trial at about this month. Like, that's what we want, but we probably can't get that.
Yeah. So there, several people put in the chat that they really like that idea, Carmen, the list that you gave. So we definitely need to try to make that happen for the next meeting. I believe our next one is at the end of May, towards the end of May. It's the 30th. Oh, no, I'm
I
sorry.
think we have one in between.
The next one is May 2nd. No, I'm sorry, that's wrong too. I saw PO and thought that was Pompeii. That's actually prom because my son goes to prom next week. The next meeting is May 9th. And it'll be at the same time. And again, we always record the session, so if you can't make it, you can always access it that way. Brian, I'm so glad you and Fernando were able to come. Just trying to make sure. Okay.
So apparently, Heather, at the next meeting, you need to open up with prom pictures since you've told us all of
We'll see. We'll see. You know, he doesn't like to get his picture taken.
this. I only have girls. They love to have their picture taken.
Yeah, no. Mine stopped a long time ago.
Probably
Yes.
if you tell him if it goes all around the world, he will like it.
Yes, Jose, this webinar is recorded, and once it's been edited, we'll put it out there for everyone to have access to. All right. If there's no additional questions, oh, I'm sorry. I'm sorry, I just wanted to, so Jose, so Pompe Alliance has a YouTube channel, and when we post our videos there, and then we have a Facebook page and a Facebook group, and so we put the link to the meeting in our Facebook page and our Facebook group, and then we also share it in some of the other Facebook groups, and I know some of the other groups, they share it too. I think Allison from Grants Giants is on here. They share it. Um, uh, um, Pompeii Warrior Foundation, Anne, who has collaborated with us on this meeting but can't be here today, she shares them. So they'll be in a lot of places for you to have access. And if you can't get it like through one of those, if you shoot Pompe Alliance an email, pompealliance@gmail.com, I can send it to you. And if you have any questions or anything you want to add that, maybe we didn't discuss today, you can also shoot me an email, pompealliance@gmail.com, and something that you have a suggestion for the next meeting. Okay. Thank you, Carmen and
Thank you.
Caitlin. Again, we appreciate I appreciate your expertise and your time. And I'm so glad to see so many people from all over the world who have joined. I hope you make it to the next meeting. And everyone have a good weekend.